All right, everybody, here we are. Project number 4, Unit Root Tip Lab. This one's a pretty fun one. I want to quickly go over the background content on mitosis, specifically on which we'll be looking at, diving into the different stages of mitosis, and then I'll walk through the lab itself. Okay, now this is a pretty common lab and pretty simple to do, and so I'll walk through all that with you guys. So to start out, let me quickly pull up my lesson plan or my PowerPoint for you guys. I'm sorry, I usually have this all set up. Okay, so here we go. Let me start this puppy. All right, so to start out, what we have is the process of cell division. We will be looking at, as it said right there, onion root, to look at and see the stages of mitosis. Onion root's a pretty good one just because it's at that stage of the onion root. So you have to get, like, if you had like a little baby onion, it's got these little stringy things hanging down from the bottom. And if you get down to the very end, that is where growth is occurring in those root tips to go and find water and other nutrients in the soil. And so if you take that, you can actually see very active cells going through active cell division and growth. And so to start out, I wanted to dive into specifically looking at chromosomes. And if you look right here, you guys, you can see that this is a chromosome right here. And it's chromosome will take, DNA will take multiple shapes. So one shape it could take is single helix, double stranded single helix or double helix here. But if you zoom in, but if you really look at it, it's wrapped around histone proteins is what these are called. They'll wrap around themselves to make what we call a nucleosome, and then these will tightly compact to make this kind of ropey structure, and then all that wound together makes what we call a chromosome. Now this looks very high depth. Again, these you can't even see it without a microscope. You would never be able to see this in a light microscope that's typically used in high school science classrooms. And so, but you can see the chromosomes when they're like this. And if you get all the DNA wrapped together, you can actually see it, and then with the naked eye, it looks like snot. And so, went into that. So when we're looking at cellular division, specifically in plants and animals, we follow this type of eukaryotic cell cycle. Okay, and so we start out in what we call G1 phase, and usually cells are always found in somewhere in this. And there's actually another part right here. If I was to draw it, that would be like right in here, and we call this G0 phase. And it's the G0 phase is where, like, if the cell is stagnant. I can't spell the stupid thing. Anyway, stagnant. It doesn't need to divide. It's happy to be where it needs to be. It can be in what we call G0 phase, which is just nothing's happening there. And then what we see is let's say that, for example, the cell is ready to divide, it will receive some sort of external stimulus or internal stimulus saying, hey, it's time to grow. It'll dive into the G1 phase, into the S phase, into the G2 phase, and then into what we call cell division or mitosis, which is what we're specifically going to be looking at. But interphase has some important qualities and stuff that happens in there that I want to talk about. So in interphase is a big, large section of time here. And in the G1 phase, we see cell growth is occurring. So the cell is getting bigger cytoplasmically to be able to allow for the cell to, when it does split, turn into two equal size cells that were the same size as the original. Also, you have to think about all the different organelles inside of the cell. We have the mitochondria, the chloroplasts, the vacuoles, the Golgi body, the ER, the ribosomes. All those things have to be duplicated and made multiples of, so that when it divides, the cell gets equal amounts. So that takes some time. Another thing that takes time is we have to replicate all that DNA so that each cell ends up with similar amounts of DNA. During the G2 phase, this is where it will prepare for mitosis. It might check DNA for repairs, for issues that might occur in that DNA process that might have occurred, and fix it. And so let's skim through that. Okay, so here we are. So the first phase is called interphase, and during interphase, again, we see these three steps, G1, S, and G2. A typical look for a cell that is in interphase would be this. And this is what I want you guys to really pay close attention to help you with the lab. So in interphase, you'll see cells. This is a plant, an onion root tip. Here's the nucleus. You can see these two to three distinct dots. That's a key identifier that these are in interphase. Not a lot has gone on. Maybe the DNA is starting to replicate. Maybe the cell's getting a little bigger to grow. It's working on one of those, but not a lot of visual change. The second step in metaphase, or in mitosis, is prophase. And in prophase, there's a couple things happening. So first of all, you'll see that the nuclear membrane will disintegrate. You cannot see a nuclear membrane in this one. You'll see that the chromosomes are starting to condense to make chromosome structure. So the DNA is condensing up to make kind of like this spaghetti string. The last thing that you'll see happen is, if you look at these, all these little lines here, these are called spindle fibers. And it's these spindle fibers that are actually causing the cell to, or it's going to attach to those chromosomes and play a very important role. Okay. Again, those are spindle fibers or microtubules. They call them multiple things. And so the next phase would be metaphase. So in metaphase, we see the microtubules or spindle fibers are pushing those chromosomes to the center. They line up at the center of what we call the metaphase plate. And when it goes to that, we see the chromosomes will start to separate apart during anaphase. Each cell will get half of that, identical copies of each other. And this is what it would look like in real life. And then we see the nuclear membrane reform around those chromosomes, and the cytoplasm starts to pinch off to the point where we have cytokinesis where it will directly pinch off. Okay, so just a quick review. We have interphase. What comes next? Prophase. What comes next? Metaphase, anaphase, telophase, cytokinesis. So when we're looking in the microscope, this is what we're going to do. We're going to, in this lab specifically, you will have some different picture samples of real life mitotic division occurring in onion root. And you're actually going to go and count how many cells there are in each phase. Okay, and so when I do this, I kind of like go up and down the rows like this, and I'll just count. So for example, going through, and I'll have a little paper or my lab report out. This one, what's it in? Prophase, or it's in interphase. What's this one in? Well, it's lined up at the center. This one's probably my metaphase. Interphase there. We see the little nucleus in the inside sitting there chilling. These ones don't really, you can't see what's going on, so I'm going to keep going. This one right here is probably some interphase going on. Interphase, 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 metaphase, right? What's this one? Prophase. Another prophase there. Oops. And so that's what we're going to do in that. Okay. Do I want to save? Don't save. All right. So when we're looking at this, is what kind of what you'll do there. There are some resources, but I kind of went through that, so you can click on that if you'd like. So for this lab, again, like I said, we will be counting how many there are. You'll be using these two samples, and so when you're doing that, these are the samples you'll use. There will be some pre-lab questions. So provide an overview of the phases of mitosis. Explain why root tips are used for observing mitosis. And I'll walk through that here in a little bit. So your lab data sheet will be right here. And so when you click on that, you'll be able to download this and type on that puppy, and it should save. I like to download this first before typing it up on this. but you guys know what you're doing there. If it doesn't like to work, though, always just get a Google Doc, copy this, paste it in, and that'll work for me, okay? So for the what you need to do is you'll just have to keep track of how many there are in each cell cycle. Ultimately, the driving question that we're trying to answer here is this: how much time does a cell spend in each of these phases? Okay, so if we're looking at all these phases right here, interphase, prophase, metaphase, anaphase, and telophase, how long does an average onion cell spend in each one of these phases? And that's what we're going to be testing. Do they spend the majority of their time in prophase, in interphase, or does it take a lot of time in telophase? So we're going to see how much time is spent in each phase with this lab. And so to do that, when you're going up and down the rows, you're just going to go through and you'll just keep a tally mark on here of how many are in interphase, how many are in prophase, metaphase, etc. Add those tally marks up to get a number, and then you'll be able to convert that into a percentage. And I'll show you how to do that here right now. So let me get my let me pull up a document that'll kind of walk you through this. So this is a lab I do with my kids at my other high school too. And this is kind of the report handout I give them. And so you can kind of follow this if you'd like. And so, for example, let's say that I count 50 cells that are in interphase. Let's say I count 10 cells here, 10 cells there, 5 cells there. Now let's go. Let's say I count 10 cells there and 5 cells here. Okay. What's my total? And so when you're doing this on your document, I would recommend adding a total. I don't like how they don't have it there, but having a total would be helpful, and I'll show you why. And so if I add this up, 50, 60, 70, 80, 85. Okay, here, just to make this really easy, let's change this to a 20. Change that to a 20. Change this to a 15. Now my total is 100, which will make this easy. All right, so if we want to get the percentage of total cells, all we have to do is we take this value, so 50, we would divide it by the total, which was 100. Well, what does that equals? Well, that's 50%. Or, not 40%. 50%, okay? And I can do this for each one. Well, this one would be 20%, all the way down, right? Now, what they've actually found is that an average onion root will spend about 24 hours to carry out the complete cell cycle. If we convert that to minutes, that's about 1,440 minutes. And so we can use this percentage. Ultimately, what the idea here is looking at these slides right here. This is a snapshot in time. All these cells are going through cell division somewhere in that process. So the more time that is spent in that phase would mean that the more cells we count. So in this, my hypothesis could be that the majority of the time is spent in interphase, because that is what the majority of my data was. Now, I would recommend when you're counting the cells to get like 200 cells. It'll make it more accurate. Okay, so like 100 from each if you wanted, and add that into your tally mark. So anyways, back to this. So looking at this, we have 24 hours. How much of that 24 hours is spent in interphase? Well, according to the amount of cells I counted, that's 50%. So 50% of this time was spent in interphase. So how would I do that? Well, I could just take this value right here, 1,440. I would multiply that by the decimal form of my percentage. So 0.50, which would then equate to 720, right? Yeah, 720 minutes. And so right there, I was able to calculate using this data what that value would be. And so you're going to do the same thing on your handout here. to calculate that time. And again, it says in here how much time they spend somewhere. Procedure. Yeah, so if you read the procedure, it tells you exactly how to do this. Again, 1,440 minutes is the average time. Take the percentage divided by 100 times it by that. What this is just a decimal form of the percentage. So right there. And so this walks you through that process that I just explained. And when you do that, you'll be able to analyze the data. Which stage of the cell cycle spend the majority of the time? So there's our scientific question that we're trying to test. Which stage of mitosis lasts the longest? Explain why using the data. Which stage of mitosis is the shortest? Use data. And then support the conclusion. So now to tell you exactly, interphase is the largest portion. Tell me why. So going back to the PowerPoint, think about what's happening in G1, GS, and G2 phase. Why would they need the majority of the time why would they need the majority of cell cycle just to prep the cell for mitosis? Okay. Anyways, I hope that made sense for you guys. And if you have any questions, do not hesitate to contact me. And this video will always be up for you to be able to watch. So watch as many times as you need to be able to carry out this lab procedure. All right, talk to you guys later.